First of all, I'd like to take a chance to thank Pavel and the members of the Laboratory of Algorithmic Biology for bringing together this group and for giving me the opportunity to address you, if only for a few minutes. I realize that I am at the end of the day, and as such, I know that you're all tired because your minds are maybe saturated with some wonderful mathematics and computational algorithms and discussions and even a little bit of philosophy I heard last night. So today I'm going to lighten it up a little bit by giving you a bit of an overview of comparative genomics in mammals. And I'll start by saying that it wasn't really that long ago, at least doesn't seem like that long ago to me, 2 decades, that your country here was weathering a rather painful transition from the Soviet era to what is now modern Russia. And during that time, some of the scientists weren't supported as well as they could have been, and this has led, of course, to some hardships for the peoples and some difficulties. And I think that the government here, at least as an American, who was awarded a Beggar Grant, I can say that what they're attempting to do is to recall outstanding Russian scientists back to the motherland to help rebuild some of the science infrastructure that has gone before. And somehow or other, they didn't notice that I wasn't a Russian, and they gave me one anyway. So, but at about the same time that the Soviet Union and the Russian transition was taking place in the rest of the world, at least in the genomics community, there was some exciting things going on. When scientists from Europe, Asia, and Americas got together and they decided that it would be useful to develop the entire sequence of the human genome. And that was a very ambitious and audacious project that was very controversial when it originally started, but the people who were leading it were clever enough to involve scientists from all over the world. And 10 years later, with a little bit of competition, a little bit of interaction, the draft genome of the human sequence appeared on the internet and also was celebrated in publications in Science and Nature. There were a lot of interesting things in those papers. Now, 11 years ago, we just celebrated the 10th anniversary of the Human Genome Project. Some of them are listed here. Some of the highlights, we counted the number of genes at about 22,000. We counted the number of base pairs at about close to 3 billion. We noticed that about half of the genome sequence was repetitive elements. We noticed, that's good. We noticed that about 98% of the genome, the human genome, was non-coding. We noticed that there was a lot, early on it was noticed there was a lot of single nucleotide polymorphisms on the order of 1 every 1,000 base pairs or so, translating to today over 10 million single nucleotide polymorphisms that have been annotated and described. We noticed structural variation. We noticed that SNPs were inherited in blocks. There were lots of other things we noticed as well. But in addition to that, there were a number of things that the human genome didn't tell us, despite all of the promise of the people who said, well, spending $2.7 billion is a great idea. We didn't know what most of the genes actually did. We didn't figure out which SNPs regulated which characters. We're still trying to do that. We didn't know where and how and why the genes function the way they do, how they came to be, why they were arranged in the way that they were. We didn't really know much about which genes were making us human and which were making a dog or a fish, which are critical and dispensable, which are dispensable, which regulatory elements function, and the special effects and influence, whatever it is, of paralogous gene families. All these things, these are just a bunch of questions that I thought up when I was making up these slides. There's many more. You can think of them as well. So basically, the sequence itself hasn't really realized its total promise. It has realized some promise, but not everything. So there's a lot more to do, and that's where computational biology comes into it, is helping us get to the stage of answering these questions. And the approach that I'm going to emphasize today is the comparative approach, which is not really a new one. It's basically been around for a long time. But as the Russian scientist Theodoros Dobzhansky, who is the person we named our center after at St. Petersburg University is well known to have said nothing in biology makes sense except in light of evolution. This is Dobzhansky here. This is one of his buddies, Ernst Mayr. Now, the power of comparative biology is not new. It was used by anatomists and surgeons in the 16th and 17th century to understand human anatomy by dissecting pigs and cats, organ metabolism, medicine, heart, muscle physiology. It's just now that we have sequence data that can be used for whole genome sequence. So what species do we pick? Well, the mammalian radiations, which is what humans are, one of 4,500 species of mammals, began about 100 million years ago when this little critter walked around underneath the feet of the dinosaurs. And when the dinosaurs disappeared about 70 million years ago, this group and its cousins radiated into the 25 or so orders of mammals that exist on Earth today. And that mammal natural history was used as a backdrop upon which the National Institutes of Health, which I worked for nearly 40 years in the United States, decided that we need to pick some species across the mammals in order to go ahead and to understand, if you will, a little bit better the organization of the human genome. So the first ones they picked were the mouse and the rat, which were in actually a single monophyletic clade or group that also included humans, this one here. And then they decided to pick a number of other species. This is a tree which illustrates the relationship between some of the mammalian species that are placental mammals. And it includes only those that NIH decided to pay for the genome sequence of. Okay? So basically, if you look at these, they were selected by a committee led by Bob Waterston, and it consisted of most of the heads of the sequencing centers in the United States. And I was on it because our group had basically produced the phylogenetic relationship of mammals, which was the platform upon which they picked it. And one of the benefits of my being on it is I've been working on cats for a long time. I was able to get the cat included in one of the species that was done. So in the next few years after these deliberations, which took place in 2003, 2004, a number of species had their whole genome sequenced, and they were published and highlighted the cover of many of the popular journals, which I'm sure you're all familiar with and have occasionally published some of your work in. So I was interested in house cats for a number of reasons. They're not important, but if any number of scientists could get up here and give the same talk I'm about to give, and if they were working on horses, they'd talk about horses. If they're working on pigs, they'd talk about pigs. If they're working on monkeys or chickens or something like that, they talk about those. Because I know a lot about cats, I'm going to spend the first 10 minutes telling you how to assemble and to put together and to annotate a genome organization in a domestic species, the house cat. But you could substitute the word cat for any of these other species and pretty much get the same talk if you wanted to. So house cats are a member of a group or a family of carnivores called Felidae. There's 37 species. They're one of the most successful predators in the history of mammals. They have survived for a long time. The only predator that's really outcompeted them has been ourselves. So now 36 out of the 37 species of Felidae are considered threatened or endangered. The one that is not is the domestic cat, of which there's close to 1 billion individuals left. Now the cats have evolved from Asia, migrated throughout the world, and they have a phylogenetic and historical relationship that's been worked out by some of the students in our laboratory, and it is that context in which I would like to discuss the genome of the domestic cat. Now, the domestic cat, is not really a species. It's basically a domesticated version of a wild species called Felis sylvestris, which is called a wildcat. And if it's in Europe, it's called the European wildcat. If it's in Asia, it's called the Asian or the Middle Eastern wildcat. And if it's in Africa, it's called the African wildcat. And the domestication process actually began about 10,000 years ago in the Middle East, which was the time that the first Neolithic humans were domesticating themselves. They were settling down to agriculture in the Middle East, and saying, we're tired of this wandering around all the time, and we're going to sit here and maybe grow some things, and we're going to fence up some of these animals. And these first geneticists actually began the process of domestication, which some people have called the lever that changed mankind. You may not believe that, but people who study it believe it. So anyhow, the domestic cat came along. It wandered into the villages. It found an opportunity to eat the garbage and the mice and dispatch the things. It basically had another feature that was useful, which is that it didn't eat the children, and therefore it didn't get thrown out by like all the other wild cats did, and eventually became invited into living rooms and onto ships and moved around with people. Veterinary medicine has described a parallel medical disease of domestic cats and dogs and other domesticated species. This listing, which is not really so informative, is simply the human hereditary diseases for which there are models described in the domestic cat. Things like spinal disease, things like diabetes, things like hemophilia, glaucoma, deafness, renal dysplasia, and other kinds of diseases are all described phenomenologically, if you will, by veterinary clinicians who are observing these diseases in cats. There's a similar list for dogs, there's a similar list for bovids and horses. But this is basically a medical reason and justification for being interested in getting a genome sequence of the house cat. In addition, there are some very interesting viruses that afflict the cats that are similar and related genetically to the viruses that inflict humans. This is feline immunodeficiency virus, which is a first cousin of HIV, of course, the AIDS virus. Okay. There's only one kind of mistake you can make here, go backwards, and I make it about every fourth time, I think. So FIV is a cat version of HIV, and understanding the interaction between this virus is an interesting subject. There's also other viruses that model human diseases. Most of the oncogenes which we use to describe and characterize human tumors were isolated first from house cats in the 70s during the virus cancer program. There's a coronavirus in cats, which is the first cousin of the SARS coronavirus, which caused this horrific outbreak in China back in 2004. There's a herpes virus, there's papillomaviruses. Cats are very susceptible to flu, avian flu, panleukopenia, which is a relative of measles, foamiviruses, and these are all things that we need to know more about. I'm just putting it up there as part of the I told you to go backwards all the time. The Feline Genome Project started by identifying a cat who would volunteer to have her genome sequenced. This is Cinnamon. She's an Abyssinian cat that lives in Columbia, Missouri, and I've been discussing informed consent with her. Now, this was several years ago, and since then, there was initially an NIH-funded light sequence, 1.8x, which was published in Genome Research. It basically listed some of the features. It was a sort of a crude assembly because 30% of the genome was missing, but nonetheless, it gave us a glimpse, if you will, of the genes and the SNPs and the other things in the Cinnamon's genome, which are interesting. A few years later, there was a light sequencing of a number of cats in order to complement the SNP discovery, which was published as a 3x, and most recently, Washington University's Genome Sequencing Center has finished up a 15x sequence, which is what I'm going to just briefly give you a little talk about so you'll know where we are. Now, the genome assembly from WashU was from Cinnamon. It was a 14x coverage with 454, and it had a number of paired end reads with different libraries of different size. It was assembled with Kebab, but has now been improved by Steve Salzberg and his colleagues at Johns Hopkins, and the N50 and the 12, which are specific measures of how big the contigs are listed in this slide right here. A number of SNPs were discovered in Cinnamon in the first light sequencing, but an interesting phenomenon happened because Cinnamon herself is a descendant of 3 separate inbreeding events in the past. Her genome is parsed into regions that are completely homozygous for SNPs, long regions, and other regions that are not. And in the regions that are not, the frequency of SNP variation is about the same as humans. It's about 1 every 1,200 base pairs or so. So if you look at the chromosomes of cinnamon, and these are the 19 chromosomes, the red regions have high variation of SNPs and the green regions don't. If you stand back from this, you'll see that about half of her genome is homozygous because she descended from 3 episodes of inbreeding. The first episode was domestication itself 10,000 years ago. The second episode was the development of the Abyssinian breed, which was about 50 years ago. And the third episode was development of a pedigree of individuals that were suffering from retinal dysplasia, which is what cinnamon has. She's blind. She was selected for whole genome sequencing not only because she cooperated, but because she was remarkably homozygous. And the people who assemble genomes don't like SNPs because they interfere with the alignment and the building of the contigs. So that's why we picked her in the first place. Today, we have about 10 million SNPs described for cinnamon. Now, in addition to that, I've had an ongoing discussion with the heads of the sequencing centers who think that if there's money for genome sequencing, that some large fraction of it, like 100% of it, should go to them. Okay? But they don't want any of it to go to other aspects of it. But I said, well, maybe we should have a little bit of it that should go to building a framework map so that the contigs and the scaffolds that you built will be able to be placed on the chromosomes in some order that represents some semblance of the way they occur in an individual. Well, they're coming around to that now. They're starting to begin to appreciate it. So there's physical map of the cat, which was built with a radiation hybrid map, which is the thing actually that anchored the human map 10 years ago. In addition to that, using a SNP array developed by Illumina, which has about 50,000 SNPs on it, we put this down on some pedigrees of cats so that we'll have a linkage map, which is now being integrated with it. So that hopefully we're not going to wind up with 50,000 markers because we don't have enough recombinations, but we might end up with 12 or 15,000 in addition to the 2,700, which would then give us a rather dense assembly of markers along a chromosome. In addition, David Schwartz is developing optical maps, and then there's talk about backends, although we haven't really gotten that way. Now, when we made up the SNP array, there was a number of SNPs that were basically characterized in a preliminary session, and the SNP array is actually quite good. That's all this slide says. Now, if we use the genome sequence, one of the things we can do, once we have an anchored map that does resemble its order on the chromosome, is to compare it to other species. Now, we've been doing this in mammals for 20 years before there was even a genome sequence, but we weren't getting the resolution that we were able to get when we have a whole genome map. And what we discover with the whole genome map is largely that, when we compare, for example, the human X, the mouse X, and the dog X with the feline X, is that all the markers across this region of the X are identical in the dog and in the human, and identical in the human and the cat. This is the cat up here, sorry. The mouse is rearranged because the mouse has a genome that's been rearranged several hundredfold relative to other species. Now, when you look at the other chromosomes, you'll see that the human has some rearrangements as indicated by these color differences, and it's just simply to say that these are the chunks. They're called syntinic segments, which are segments where the markers, the reciprocal best matches across the two genomes, are in the same order in species A versus species B. And when they change, you invert them or you translocate them, and you use whatever names you want for that exercise. So basically with this, we worked with Pavel and his colleague Glenn Tessler over the last few years to annotate these things a little bit better. And one of the things they've come up with is a comparison which does what we used to do on the back of an envelope with a computer. And that is to take the human genome and to color it according to each chromosome, and then ask, well, if we go to the cat, where do the sections of the genome in chromosome 1 wind up, and then color them as such according to the cat chromosomes. Then do the same thing with a mouse and with some of the other species, and then use the algorithms that specifically maximize the quality control of the mapping, which is a big problem. You know, that is to say, a lot of the mapping, there's a lot of mapping errors, just like there's sequencing errors, and so you basically throw away things where there's like one marker and it doesn't give you the right answer, and then you basically come up with a parsimonious interpretation of what all these differences mean and try to come up with a way of understanding across radiations. And a couple of years ago, Bill Murphy, Harris Lewin, and I, and so forth, we repeated this thing a little bit, and we did it with a series of species for which we had adequate maps and genome sequences. And this is roughly an evolutionary tree relating some human primates, some rodents, and some carnivores, and it just simply counts up the number of exchanges, both between chromosomes, call them translocations, or within chromosomes, call them inversions, that occurred on the different lineages. And what you can see, for example, is that the number of translocations on the cat between the ancestor 100 million years ago and today is amazingly small. It's less than 10. The same is true with humans. It's like 9. The translocations are 4 times higher, 37 and 42, but the number of exchanges are T9c. This means that the number of scissor cuts it takes to rearrange the chromosomes of cat into human is really quite few, on the order of 15 or 20 when you just count the translocations. And this is all, and this basically supports what the chromosome painting data had shown, and so forth. So this but on the other hand, it's also interesting to note that some lineages, like the dog, have a lot of translocations, and this is true as well. And this, again, reinforced something that we'd been seeing a long time with chromosome painting, which is that most of the mammalian genome is pretty conserved. It has a little bit of exchanges go on. But there are episodes in some groups where the genome gets reshuffled like a poker deck. And those include the mice and the rodents, include the dogs, it includes the bears, and includes the gibbons. The others are pretty conserved. So some of the take-home lessons from the most recent analysis, which we published in Science 2 or 3 years ago, was that the translocations and inversions are balanced. Let me show you what I mean by that. When you have a low number of rearrangements, you have a high number of inversions. When you have a high number of translocations, you have fewer. It's almost as if the number of breaks is restricted. Not how they break, but the number. I have no idea why that is, but that's what we see. Okay? And these kinds of things are coming up, raising new questions, like the problems we were discussing last night. So, other things we saw is that a lot of the breakpoints are reused. There are regions that can stand it in evolution. Another conclusion was that evolutionary rates vary. Some lineages, they're slow, others they're 20-fold higher. Reuse sites are clustered near the centromeres. And here's an interesting one. The gene density is enriched around the evolutionary breakpoints. That's counterintuitive, but that's what we say. This thing is controversial, but we believe that the data are rich enough to at least suggest it. The segmental duplications in humans are enriched in the breakpoints and pose a mechanism for breakpoint exchange. It could be that when there is a copy number variation, that breakpoint exchange might be induced and more common. We don't know that's true, but it's a guess we made based upon the data. ESTs, we have a number of libraries that were sent to WashU, 10 cat tissues, a number of transcripts, and these are of course being used for the ensemble gene identification and tissue distribution of genes, the splice forms, and all the things you do with RNA. Now, this is basically a listing of some of the features of the genome that we call the annotation. These are the things that you look for in genome sequencing and assembly and annotation paper. And most of them have been done in the house cats, including the assembly to contigs and supercontigs, the annotation of the genes, the identification of conserved sequences between other species, the homologous synteny blocks, which I talked about for a minute, and misspelled, repeats, GC content, then the SNPs, the microsatellites, the primers for those, the deletion insertion, homozygous segments like I talked about, microRNAs, CPG islands. We had a back library of the whole major histocompatibility complex, which we compared to the assembled one. Nuclear mitochondrial DNA pseudogenes, or pneumites, endogenous retroviruses, and we built a browser. We called it the Genome Annotation Region Field, or Garfield, and we got permission from Jim Davis to use that. So this is the same list. I won't go through it again. Some of the things that happen is genes for many of the different diseases and phenotypes have been worked out by some of the people here. You're going to read in Science in the next month or so the mutation that's responsible for changing cheetah into a king cheetah, which is a different pattern. The kinds of applications that come from the genome sequences are discussed regularly at a conference that goes on every 2 years called the International Advances in Feline and Canine Genomics and Inherited Diseases. The last one was in Visby, Sweden. Before that, we held it in Baltimore, and before that, it was in Saint-Malo, France. It's sort of exciting because we're seeing the whole thing happen. Now, imagine if you could take what I've just said in the last 15 minutes about the cat and apply it to your favorite species. You could. Let me explain that my friends Oliver Ryder and David Hausler and I, Ollie runs the frozen zoo at the San Diego Zoo, which is a large collection of frozen specimens of vertebrates and invertebrates. David is the director of the UC Santa Cruz Genome Browser, which has a lot of comparative genomic sequences. We were sitting around thinking what you're thinking. It costs, the human genome cost about $3 billion. But today, with the high throughput sequencing that we're able to do, I mean, even without having Pac Biosciences work, we still have very, very low prices for getting high coverage of a genome the size of humans. The reagents cost less than $10,000. They're dropping to $5,000. You could walk into the San Diego Illumina place, give a guy some DNA, and they'll do the sequence for $4,000. So we were thinking that, well, in 2006, NIH and several people who were involved in funding comparative genomics said, you know, we're not going to do any more animals, we're just going to do humans. Because we don't know how to assemble them de novo, and we got the Cancer Genome Project to work, and we got to look at polymorphisms, and we got to solve human diseases. I didn't like that, David didn't like it, and Ollie didn't like it. Because we knew that the price of sequencing had dropped so much that we could basically think about sequencing everything that we could catch. So for want of a better idea, we decided that we'd try to see if we could gather together the resources and the people to start organizing, planning, and cooperating on sequencing not just 25 mammals, but 10,000 species of vertebrates. We called it Genome 10K. It rolls off the tongue. We got a little seed funding from a genetics organization. We had a meeting in Santa Cruz. And we brought together not the smartest genome scientists or computer biologists I knew. No, we didn't bring them. We brought in people who'd spent their lives collecting specimens that had freezers full of birds, of reptiles, of amphibians, people that look under rocks in strange places for species. And we said, come to this meeting and talk to us about making your specimens available, the way Cinnamon did, for a genome sequencing if we can get it organized and find the funding. We had a long discussion. It started out actually in this saloon in Santa Cruz, and there was a lot of argument. Well, you know, these aren't going to be voucher specimens. They're not going to be perfect, and who's going to keep control of this, and who's going to pick them, and well, what's the criterion going to be, and all kinds of discussions. And this went on over a couple of beers. And then the next morning, David got up and he said, look, okay, we're going to break you fellows up and ladies up into groups. There's the group that works on mammals, the group that works on birds, the group that works on reptiles, amphibians, and fish. And we're going to ask you to look in your databases. And then we're going to ask you to pick out all the specimens that you actually know about, that you could put your hands on, that we could get you to donate for whole genome sequencing if we paid for it. In 4 days, they had produced a database which, out of the 60,000 vertebrate species, they had represented at that conference 17,000. That 17,000 was the beginning of Genome 10K. We published a white paper on that, and we sort of announced that we were going to move forward trying to gather the specimens. And we were roughly thinking that we might get 3,000 of the 5,000 mammals. Now, half of mammals are rodents, so we didn't want to do every rodent that runs around, but we want to do everything else. A number of birds, reptiles, amphibians, and so forth. We picked species that were attractive, species that people cared about, species with biological communities. And we even set up explicit criteria for picking species, such as phylogenetic breadth across the radiation of mammals, of amphibians. Is there a scientific community around it? Is there a context or use or a biological question? Is it a popular species that has use, like a giant panda or a condor? Are there interesting biological questions? And importantly, can we get a specimen? I mean, the ideal sample, we set up criteria. These working groups not only pick species, they thought about these things, and they published a series of criteria. We want vouchered specimens. We'd like to have cell lines so we can make iPS cells. We'd like to have access to fresh material. You need a lot of DNA, almost a milligram. You need a permit trail so I don't go to jail. We need a heterogametic sex when identified so we can get the Y chromosome. And we need the possibility of physical mapping, a map. And if it's and the possibility of trios, so we can look at transmission. All these things were written down. Not all of them have it, but that basically was the idea, was to see where we're going. This young fellow, Wang Jun, got in touch with us. He says, I want to be part of this because my colleagues and I, we manage a pretty big genome sequencing center, the Beijing Genomics Institute. Henry Yang started it. It sequenced the first 1% of the human genome. We set up a memorandum of understanding with BGI, as we did with the Broad, as we did with the Texas Baylor. And I said, if you did the first 1% of the human genome, this is Li Ringyi, he wrote SOAP de Novo, the assembly algorithm. I said, if you did the first 1% of the human genome, how about doing the first 1% of Genome 10K, 100 species on your nickel? And then what we'll do is we'll provide you with a community, with the specimens, with some recommendations. They asked me to sit on their advisory board, and they became full partners with Genome 10K. All of a sudden, we had a little money, okay? We had 4,000 people working in Shenzhen that were dedicated to sequencing of new genome species and to providing material for all of you to help us annotate. Okay? We started with 24 or 32 species when NIH shut the door. NSF told us the same thing. They said, we can't afford it, it's too expensive. Since then, in 5 years, Genome 10K is about 3 years old. There's been about 110 species that we've had nothing to do about sequenced. BGI has, BGI has picked, what are we doing here, boss? Okay, I'm sorry. BGI has picked 100 with the community, the G10K community. The Broad has picked another 100 with the the community. Baylor, 7 primate species altogether. There's now 318. That's a 10-fold increase over where we were when we started. Let me tell you a little bit about them. The first species we picked were fishes and amphibians and non-avian. These are the numbers. These lists are not impressive, but when you stand back from it all, I'm blown away at how the community has basically embraced this idea of getting these sequences done. Now, fish, half of vertebrates, 30,000 species are fish. We're all fish. We all descended from fish. They all came out of the ocean. And we don't know an awful lot about many of the species, except to have them described. But BGI picked 29 species. They completed 10. They have 14 in progress that'll be completed this year, and another 5. They're just waiting for us to get them the specimens. This is the phylogenetic tree of some of the species as of 2 years ago. It was much less than that a few years ago. With the new species added, you can see that the phylogenetic breadth of this radiation is starting to get filled in pretty well. These are all committed, paid for species genome projects. Amphibians. Well, there were 10 or so at our last meeting we got together. We wanted to get at least 1. There's 2 in project. There's 7 that are waiting to go. These need to get moving. This is the phylogenetic tree and shows you where the species fit. It's not as good as the fish, but it's coming. Non-avian reptiles. It's called non-avian because birds are really reptiles. We know that now. There's 12 ready to be sequenced and this many going on and completed in progress. In birds have gone crazy because Goji Zhang and some of the group from BGI and some of the other places just love birds. They want to sequence all 10,000 of them, actually, and that's what they're talking about. They basically have gone ahead and finished the sequencing of 19, and they've got another 30 that they've just recently finished as well. And they produced a phylogenetic tree of all the species that they sequenced, and it's being led by these 3 guys right here. This is all part of the G10K projects. Mammals, well, as I said, there's 4,500 mammals. There's 35 that were to be sequenced, about to be, and most of them are done. Excuse me. Mammals. Okay. In fact, a summary of where the mammals are, taking the G10K, the BGI, the sequencing centers, but there's also a lot of small universities or big universities that have one sequencer who have said, we want to do this one, and they do. And so today, we're now at the stage where I can give you a list on our website, genome10k.org, of 195 mammals that have been included that are spoken for, that are either done or will be done within the next year or so. G10K has had other advances that I want to tell you about. I didn't do all this, but I watched it happen under the umbrella of the Genome 10K Consortium of Scientists. This is the white paper we published this 3 years ago announcing Genome 10K. This is a tissue sampling methods for quality control of the samples. How good are they? It's coming out this year or just came out. This is a story about discussing just the fishes of Genome 10K. This is the study of the assembly, not a very good one, of the giant panda genome, the first G10K success. The naked mole rat has come out last year as well. The yak came out and shown it is adapted to high altitude. There are some work going on with several monkey species, including macaques. There was a competition that came out at the last G10K meeting and was handled by, which was called the Assemblathon. What the Assemblathon is a frank admission by the community of computer scientists who design assemblers that we're not sure which is the best one for assembling a vertebrate genome. So let's try it out. They organized about 18 or 20 centers, and they said, let's sequence a synthetic genome. This was the Assemblathon. So Hausler's group synthesized a synthetic genome, a model, if you will, and they had 17 or 18 centers contribute to it, including the Broad, including BGI, including Nubler, including Kabag, including most of the assemblers. And what they discovered was that by the criterion they used the best assemblers were Soap de Novo from BGI, All-Pass LG from the Broad, string graph assembler from Cambridge, and then they said, well, you can't really believe a synthetic genome. We should do a real genome. So they did. And they basically took the sequences from a parrot, a cichlid, and a snake, and Assemblathon 2 is an attempt to evaluate these. This is published. This is not. It's still being analyzed. But it's exciting because the cooperation is going on between your community, the biological community, to try to see what can be done. It all started with collections like Ollie's at the Frozen Zoo, like mine at NIH, and like those of the other consortium of scientists. And these need to be collected and encouraged at a very high level of quality. Last year, it was determined that there was a little bit of uncertainty about aligning genomes between different species. So a competition like the Assemblathon was conceived by Hausler and his group to bring up what he's calling the Alignathon. And we're going to get the results of that at the next meeting of Genome 10K, which is going to be next April in Fort Lauderdale, Florida. You're all welcome to come. In fact, the more of you come, the better we will be equipped to handle 10,000 species of genomes. Offspring, things that came out of Genome 10K that I didn't think of. Insect 5K. A fledgling group of characters who says, well, you know, there's more insects than there are anything. But there's a lot of insects that are very important to human health and agriculture. Let's pick them out and let's do a genome sequence. We visited with them, we cooperate with them, we're kind of like first cousins. They've had a meeting, they've even got NSF funding. Global Invertebrate Genomics Alliance. Invertebrates. Not insects, not vertebrates, coral reefs, sponges, these sorts of things. This thing's just starting. They're basically hosting the Genome 10K meeting in Fort Lauderdale, Joe Lopez and company. Repertoire 10K. This is an attempt to sequence 10,000 people for just their immune response genes, the T-cell receptors, the MHC, cytokines, and others. It's an exciting project, and I'm on the board of this one too. Plants, 1,000 genome plant projects. This is something BGI put together because nobody else did. But basically, they'd like to sequence 1,000 plants in the next 5 years. This one wasn't spawned by G10K. They came out first. I've been influenced by them. I'm the chairman of their advisory board, their scientific advisory board. What this is an attempt by scientists and naturalists throughout the world to put a DNA signature, a mitochondrial DNA, a CO2 gene, 600 base pairs, 620 base pairs, on every species on Earth, so that we can really categorize them with the equivalent of the name, address, and social security number. It's basically 5 or 6 years old. The number of species across the world that have been volunteered for DNA barcoding is here. They published a few hundred papers. You can find them on the web. They have centers in China, in the United States, in South America, and Asia. It's an exciting prospect, but I have learned from them how to coordinate, or at least the mistakes and the successes that they've had in organizing international projects like this. A new journal was launched by BGI last month called Gigascience, which is a journal that is meant to house these large data sets, these large genome sequencing data sets, genome-wide association studies, transcriptomes, proteomics, in addition to having a paper attached to it. So they've got a cloud computing platform that they are basically putting out, and they are publishing short reports like abstracts of what the paper is, but the data is deposited for public viewing. One of the first examples of this is the Darwin's finch was sequenced recently by BGI and freely released on their platform in the spirit of open access to the sequences. And the whole idea, again, is to make it available for everybody. One of the benefits of all this is, as somebody who loves his cats, we've enjoyed seeing the sequence of the African cheetah, the African lion, and the Asian tiger. And we're in the process of taking a look at these with some of our people here at the Dubzhansky Center. I hope to get some of Pavel's group here at the algorithm biology involved in this, sort of get practicing, and then eventually start training workshops where people will learn how to assemble and annotate these genomes, as I said. Now, I didn't do any of this work. It was all done by other people like Warren Johnson, Marilyn Raymond, Carlos Driscoll in our lab, Joan Pontius, Doug Smith, Sante Gear, Justin Lin-Bateau, some of the folks at the different centers that are going on. And the 12X, it's been a much bigger group. This group is working together to finish up the annotation of the house cats so that we can go on to the other species. On Genome 10K, this is just the fish group, but there's also a slide like this for all the other groups as well. At G10K, these are the three. Excuse me. These are the three directors who I have worked with to try to move forward the accomplishment of these genome sequencing in exchange for what Genome 10K is offering. These are the best looking pictures I could find of the founders of Genome 10K. They don't really look this good, but that's Ali Ryder, that's Hausler, that's me. Okay, thank you very much. Yes, Max.
What is happening or has happened with sequence of genomes? Are they assembled by some assembler or just keep them?
It depends on who does it, and it depends on who has access to it. I think there's a few that just the raw reads are shoved into a database. Most of them, when something is done at BGI, it immediately gets assembled with SOAP de novo with some of the informatics people. At the Broad, it gets assembled with all pass, and then there's a then when they release it, other people get to work on it.
Do they still keep the raw reads?
Yes. Yes. That's the idea at BGI for sure. It's not the idea at NCBI, and it's difficult to get them out of the sequencing centers. But I basically, one of my jobs is to try to make them keep the raw reads so that new assemblers can get at them.
Okay.
I don't always succeed, but I've told them that's what I think the community wants.
More questions?
Yes, sir.
So, about genome comparison and mutation, does it make sense to compare genomes?
A little bit louder, please.
You told about genome comparison and genome annotation. And
what's all about genome comparison?
Annotate genomes in order to compare them.
Yeah. I'm sorry, what was the question? The last part? It's all about, you said, and then what'd you say?
Is it necessary to annotate genomes to compare them, or we just compare genomes based on bare sequences?
No, it's not necessary, because by annotation I mean 20 different things, and other people mean 20 other different things. So in order to compare genome organization between different species, the only thing you need is to be sure that you have a quality control on chimerism in your contigs and your scaffolds, and that you have some sort of framework map upon which to lay these down. Does that answer your question?
Yeah.
Okay. Yes, sir.
So I've worked on a couple of genome projects, so it tells. I mean, so many of them. There are so many of them, and it seems that there is lots of sequences, but the actual science coming out of that is lagging. There's problems, annotation. Like if you just look at the, let's say, chicken genome, it's difficult to even get decent protein genes, protein coding genes annotation. So are there any efforts to boost up somehow the activity analysis side?
You sound like the editors of Nature and Science. Where's the science there? You know, when you submit this stuff. And of course they're right, and you're right. There are opportunities that are not fulfilled with many of the genome sequences that we have. And what's limited is the cooperation of people who are talented enough to do this in each of these species. And that's where we need to go. We need to get more people who have some of the skills that the computational biologists have in the same room with the people who are the users, who want to see what the genomes are, what the endogenous retroviruses are, or what the endogenous, what their MHC looks like, or what the other kinds of biological questions are. But unfortunately, it's such a harrowing issue to sequence a genome that's 3 gigabases long, perform the QC, and get it to the stage where you're actually not embarrassed to show it to anybody, that these people try to publish it at that stage and they run into what you're saying. I'm not so worried, having been around a long time, that we're not going to get there with some of these sequences. I personally think that anybody who sequences a genome should do the best they can and publish it and release it, because that way the rest of us can get their hands on it regardless of what they think they've discovered. And so, but what we do in Genome 10K is we have said, look, if you're going to be part of the Genome 10K consortium, then you should know where we're trying to head with respect to annotation. We'd like to have the chimeras eliminated from the contigs and from the scaffolds. We'd like to have the SNPs annotated. We'd like to have at least 2 approaches for gene identification, because we think that the gene prediction algorithms make mistakes. Maybe a comparative analysis as well would be good. And there are tools out there that you can do that with. And then we'd also like to have an attempt to get at some of the features of the genome that are going to be useful. You may not discover signatures of selection that explain everything, but you use the chicken genome. I mean, the chicken genome is a little bit of a difficulty, but there was a very, very interesting paper about a signature of selection in the chicken genome that came out in Nature a year or so ago from Leif Anderson. And what he found was that this region had a gene in it that he suggested was the gene that allowed domestication. And there's a lot of people looking for domestication genes in these things as well. I'm not sure he discovered the real one and only gene for domestication, but he tried. So.